All right, let's get started. So, hello again, everybody, wherever you are. Welcome to the 2021 GemPharmatech webinar series for June, marked by the summer solstice, which was on the 20th of June. It is already hot and humid where I am, and I hope you will have a pleasant weather to enjoy after the webinar, wherever you are. For today's webinar, we have invited the CEO of GemPharmatech US, Dr. Mark Moore, to join us, who will share an introductory presentation of the company and his vision in leading GPT going forward. As a brief intro to Mark, Dr. Moore completed his undergraduate study as a biochemistry major at Princeton University and received his PhD in molecular biology and immunology from Brandeis University. Shortly afterwards, he embarked on his journey of entrepreneurship, first as a senior scientist at Genetech, and then as CEO, co-founder at Deltagen. Following these milestones, Dr. Moore played a major role in the NIH initiative, the Knockout Mouse Project, KOMP, and served as executive director for the International Mouse Phenotyping Consortium, IMPC. For anyone in the field of genetically engineered mouse models, whether as a user or a model provider from academia or industry, both KOMP and IMPC are familiar names. Before joining GemPharmatech, Dr. Moore was CEO at LifeEdit and Glialogics. Without further ado, welcome, Mark. The stage is yours.
Thank you, Zhiying. I appreciate the introduction. It's a pleasure to be here speaking with everyone. Kind of odd, we did notice we had several hundred people registered from Germany, but for some reason they're not on the call right now. And my dear colleague, Pradeep, I believe is the only Englishman not in a pub. So my apologies to any of you who don't follow soccer, and my deepest apologies to my colleague, Jan, who's on the call, who's German. So please forgive the joke, Jan. So welcome to GemPharmatech. I'm delighted to tell you more about the company and a bit, and I will share my screen and start a brief presentation. So what I wanted to do is give those of you who are joining just a brief introduction to not just GemPharmatech, but also a brief history of the vision of genetically engineered mouse models and where it has started and where it's quite exciting to where it has progressed today. And again, I'm sure many of you obviously on this call have been working with mouse models, but one thing that I had to do in my role with the Knockout Mouse Project and the IMPC is to tell many people, from politicians, legislators, to lay people, the importance of animal research and particularly genetically engineered mouse models. And in fact, one key note of this is that the vast majority of Nobel Prizes in physiology and medicine have utilized animal models. In fact, over 222 award recipients have received those. So I think that's part of my messaging to show that animal models clearly have played an instrumental role in furthering medical research, physiology, and improving the therapeutic treatments we have for people going forward. But quite honestly, I believe this has just been the beginning, because with new technologies, these models are only getting better. Going back in time, circa 2005, shortly after the human genome was sequenced and the mouse genome, which by the way had almost an equivalent level of investment by the US NIH to complete within 2 years of the Human Genome Project, the thought there was, what do we do now? We now have sequenced these genomes. At that time, spent over $5 billion US for the first drafts of the human and mouse genome. But now what we had discovered were over 25,000 genes, down from the original estimate of about 150,000. But we only had a glimmer of what maybe 2,000 of these did. So what does one do? So Francis Collins, who at that time was the director of the Human Genome Project, assembled a group in Banbury Conference Center in New York State, where we discussed what we should do with these. And a decision was reached by a group of academics, some legislators, as well as people from industry, to use reverse genetics, to go from the mouse genome and exploit the exciting technology of embryonic stem cells and create a knockout for every gene in the mouse genome, and make these freely available, not freely, but very cheaply available to academic researchers as well as to people from industry to start exploring the genome by using reverse genetics. And I was fortunate enough to be one of the leaders of this project, and it was quite successful, where we were able to, within a span of 5 years, using ESLs, create a library of all of these embryonic stem cells. Just 5 short years after that, we launched an initiative called the International Mouse Phenotyping Consortium, because we realized that next tier in the pyramid, moving up, was going to be far more expensive, much more labor intensive than making the ESLs. So we launched a program. A lot of this was spearheaded by Steve Brown at MRC Harwell and Alan Bradley, who was then the director of the Sanger Center, to initiate the International Mouse Phenotyping Consortium, of which I was asked to be the executive director. And in a space of just a couple of years, we were able to assemble a team of over 16 different research institutions and 13 companies who would take the embryonic stem cells, receive funding from their parent organizations, turn the ESLs into mice, and then study the gene function by looking at a phenotypic profile of these. So it was a very exciting program, again, with a tremendous number of great collaborators throughout the world. And this was one point where I met Professor Gao, who was the leader of MARC in Nanjing, China, who later, and you'll hear more about this from Pradeep, became the chairman and founder of GemPharmatech, sort of a spinoff of all of these efforts. This program was going extremely well until everything changed. We had a great collection of ESLs. We were churning them out, making mice, but it was still a slow and expensive process. And then CRISPR came out. And as you can see the quote here, everything changed really fast. And in fact, it's hard to think of anything that changed faster in the field of mouse genetics than this, and how CRISPR now not only sped things up, but allowed us to skip ESL work and go directly into a mouse zygote. This saved us more than 2/3 of the cost of these projects, and also essentially eliminated failure, because virtually everything could be done using CRISPR. But CRISPR not only allowed us to do knockouts, it allowed us to do much more sophisticated things, which we always dreamed of engineering the genome, to humanize mice, to put in point mutations, to very efficiently put in LOXP sites where you can have a conditional knockout, again, very efficiently and much more cost effectively. And Pradeep will tell you more about that, which is the crux of today's seminar. And so just to tell you a little bit about GemPharmatech and the directions we're going, we invite you to visit our website. We are developing a vast resource of mouse models, not just CKO and the KO lines that you will hear more about today, but humanized lines, immunodeficient lines, as well as doing custom engineering using the Cas9 CRISPR system, and of course adding additional downstream phenotyping, for which we have quite a bit of experience, and preclinical studies. So this is an incredibly exciting time, and in fact, as you see, this has changed tremendously in 15 years, and in fact, just the past 8 years. So where do we see this going? And I think one major level is going to be exploratory research, of which we believe the KOAP mice are going to be critical, where now there's the library of not just ESLs, but mice, not just knockouts, but conditional ready for every gene in the mouse genome. This is going to be a tremendous research tool. Moving further from the bench, closer to the clinic, are going to be humanized and rare disease models, because these technologies have really allowed us an unparalleled access to these types of developments. The speed of discovery has been accelerated, but I think more importantly, we can now develop better mouse models that are of more true clinical relevance, whether it's going to be compound mutations to make better models for Alzheimer's, or humanization of immuno-oncology pathways for which you can test your novel therapeutic. Again, I believe this is an incredibly exciting time, and as exciting as the last 20 years, which I've just blown through in about 1.5 minutes, I think the next 20 years are going to be even more important. And I think that the breakthrough technologies that we have and the ability to do this is just unparalleled. And it's going to be a very exciting time to be a research scientist. So with that, I will stop and turn this over to Pradeep for the rest of the presentation. But throughout the presentation, feel free to send us messages with any questions, and we'll try to address them either during the presentation or at the end. So again, thank you very much for joining this webinar today.
Well, hope everyone can hear me and can see the screen. Hello from Wembley. I'm actually 3 minutes away from the stadium. It's a very happy London. It's a very happy England. Apologies for any Germans joining us today. Tough luck. That's life. We're going to keep going, but hopefully we can cheer you up today with some knockout all project data, some exciting things happening here at GemPharmatech, and how we can contribute to everyone and to their mice dreams and portfolios, and how we can take along their project as well. So today's seminar, I'll probably talk about what the Knockout All project is, and why we're doing it, and how we're doing it, essential questions. So what is the KOAP? It literally stands for the Knockout All Mouse project here at GemPharmatech. And it's a very ambitious goal of us set a couple of years ago, where we are now trying to make conditional knockouts and knockout mice for all 22,951 protein coding genes. We think, and we know actually this is very doable. We've gone through a lot already, and we are hoping, well, we know we're going to become the largest resource bank in the world soon, if not already. So I wanted to put this kind of slide in. I've been very lucky during my postdoc to travel the world and go to different kind of markets, shops, shopping malls, and I have a huge sweet tooth. So I like to try all these sweets. And that's exactly what the KOP is about. It's that Western saying of be a kid in a sweet shop, that kind of mentality. If you're a researcher, if you want to come in and say, you know what, I have this interesting set of data from RNA sequencing, I've got these genes, but what can I do? Well, you can be a kid in a sweet shop and contact GemPharmatech and say, I got these genes, what can you do? And we'll probably say we got all of them on shelf, fluxed or knocked out for your taste, and we can actually work with you if we don't have it to make a new one as well. So it's really allowing the researchers, because most of the GemPharmatech family are researchers themselves, to be a kid in a sweet shop. So we want to enable science to flourish, and we want to enable you to flourish as well. So we already actually enable a lot of people to flourish, because if you look at our existing KOAP mouse database and our customers, they've gone on to publish over 170 publications. I think it might be a bit more now. And these are in top journals like Cell, Science, Nature, and PNAS, and so forth. So our mice are available, but they're also validated and they're also published as well. So that's good to know. And that's reassuring for all of us joining us as well. So how is the KOP achieved? How do we actually do the Knockout All project? So there's three actual ways. We have the spontaneous mutation, just to go over the science. We have the random induced mutation, and we have gene modification targeting, which we use a lot. So gene modification targeting, if you split that up, you have the knockout, the KO and the CKO. You have the knock-in, where we add gene and point mutation reporters. And then you have complex modifications, as Mark mentioned, where you have multiple targets, humanized modifications. And then you have what the transgenic TG stands for, is overexpression of gene fragments by random insertions. I put this up here because I wanted to show you all that this GemPharmatech can do all, and all these gene modifications. So if you say knock-in or complex modifications, we can do that as well. If you come up to us and say we want 3 different targets of humanized genes, we can do that as well. By the way, we have a very extensive humanized portfolio in our oncology model, oncology portfolio, I should say, where we have single, double, and triple target humanized genes. But for today's talk, we'll concentrate on this, which is the knockout all kind of gene modifications, where we use KO and conditional knockouts. So if you look at the history a bit, we can see that the Nobel Prize in 2007 was won by these gentlemen for actually making this gene modification mice. And they used a very classical ES kind of technique where they culture the cells, then they used the modified gene vectors and so forth to inject blastocytes, and then they make the mice themselves. This is actually gone on over the years, and we had different genome technologies like Talon's ZFN, and lately the craze in CRISPR-Cas9. When we saw CRISPR-Cas9, we actually jumped on it as a company. We're one of the first to do so. We actually used these different techniques in ways of, just as I mentioned, transferring and re-derived mice. But we actually finally settled on CRISPR-Cas9 purely because, as Mark mentioned, it has reduced timelines, as you can see here. And also it allows us to have different various backgrounds be made as well. So it saves huge time for us and you, the consumer, particularly in the first stage where you make these constructs from the traditional technique with 4 to 16 weeks, now it's 2 to 6 weeks. So that's a major leap forward. And if you look at our data in terms of mouse model generation, if you look at the birth rate and also the germline rate as well, you can see in the CRISPR-Cas9 conditional knockout is 12%. And also if you look at a conventional knockout, it's 20% birth rate. And the knock-in, we actually have data as well, is at 12%, which are very good numbers. And if you look at the germline rate itself, it's actually 2.4, 7.5 respectively, and 2%. So we can see why we use the CRISPR-Cas9 technology and why we stuck with it to do most of our models. And what's exciting is, if I want to give you an update, we started only in 2019, but in 2021, currently our gene targeting and our work has now completed 10,100 of these genes. So that's very exciting, targeted. So we're going on a rapid pace. It's unbelievable what the team and technical team can do. We can generate around 5,000 or more of these mice per year, plus the other mice as well that we look after and also plan to do. So it's a very exciting time for us, and it's going to be an exciting time for you because you have a very diverse portfolio to access. So the current status is that we just don't go by, oh, we think this is going to sell, or we think this may be good. No, we go by what our customers want, and what the demand is and what the scientists need. So if we look at our strains, we can see that so far our knockout, constitutional knockout KO strains are numbering 8,000. Conditional knockouts are around 5,000. And this overall, the latest figure I just pulled out from the technical team, 10,100. That might not add up, but this is what we're having so far as of yesterday. So that's quite nice to know. And we actually target everything and anything. So as we can see here, GPCR receptors are quite high up, but we can also go to the cyclins. We can also target ion channels. We can even ask, if you come to us and say, you know what, we want this, we can also do that for you as well in terms of any organelle or any cell parts or anything you need. And talking about current status, you can see here, if you look at intercellular components, so far we actually targeted a wide range. This is actually based on customer demand and want. So if you need nucleus type proteins be knocked out or gene function studies to be done, we got nearly 5,000 of those. And we can also go with super smooth endoplasmic reticulum or mitochondrial knockout. So really, you're that kid in that sweet shop. The world's your oyster, and you can actually tell us what you need, and we'll probably have it, or you can actually demand and we serve. And to look at our portfolio even further and look about fields, so we looked at cellular components, but if you look at physiological fields, we can see that by far we have a huge portfolio in the immune system, which is we do very well in. We have a lot of expertise in our technical team for the immune system, but that doesn't stop us as well. So we can also go into very unique niches like the hearing, auditory niches where we have actually quite unique demands. So that's quite good to know that we actually go into niches that others don't even touch, but we actually go and help our researchers. We also have a huge portfolio in aging as well. And we have the capability of actually making these conditional knockouts and aging them as well. So that's something for you guys to remember as well. Again, some other things to highlight, we look at the cardiovascular system as well, which is quite productive for us, and also the neurological system. So field wise, we have no limits. Just come and talk to us and probably we will help you. So we want to talk now about how we covered what and how the knockout all project is, but how can you get involved? How can you make orders? So if you go to GemPharmatech.com, our website is just like a normal shopping kind of website where you have the shopping cart. You can actually search the model or the item you're interested in, and it gives you a very extensive availability list, detailed strategy on what we do and what we offer. And you can just add to cart and inquire, and then one of our sales teams across the world will get back to you in a very quick time. And one of the cool things about this website, what I really enjoyed when I first started working here, is that you can actually break this down in terms of your specialty, your pathways that you're looking at, or your research niche. So you can really look at different ways and how you can get these knockout mice. For example, if you click on AMPK pathway, it kind of gives you all the signaling pathways associated with AMPK, and you can actually click on the protein in the pathway, and then it will bring out the available knockout mice. So quite cool, quite interactive function of the website, and it's quite convenient as well for researchers to look into what's available and how quickly they can obtain it. We also don't just stop by giving you the mice details and what's available, and we don't like wash our hands. We actually have very good pre and post sales teams and functions, and also support. If you look at pre-sales, we actually give a very comprehensive software for strategy design. If you don't have a strategy, we can design it for you. And if we have the mice, we actually provide you all the schematic diagrams and strategies and how we actually make these mice and what the modifications are. We don't stop there. We also do sequencings for you. We actually look at things like the sgRNA primer details and donor sequencing as well. So we're quite comprehensive in what we show our researchers. And also here's a kind of a cool optimization slide here that shows us how comprehensive and how thorough we are in terms of what we do, from the start, from removing sticky ends of the PCR all the way to checking dephosphorylation, your DTA double oligo, and also looking at even your ssDNA. So we do all these things very comprehensively, and we validate pre and post for you. And again, there's another QC standard optimization where you can see how we look at tandem repeats ratios, and you can see how far they drop. And we also look at first pass yield, and we look at the flowering rate and also the positive rate as well. So again, it's nothing that we do randomly. We actually take our time, validate these mice. We talk to our customers. We have their demands as well, and we actually give what you guys want, and we actually validate very thoroughly before we give it out to customers. Those are the knockout mice, but with any conditional knockouts, you need CRE mice, and we have you covered on that as well. We have a very extensive CRE library that stretches from the immune system to the nervous system to the bone and muscle, a very unique niches. Our CRE strains now from last October starts just below 200, but I have been informed there are much more now. We also have CREs that we need researchers to validate and help us on. So please, if you have a CRE in mind, or if you think we know a CRE that we have and you want to work with this, please tell us. and we're more than happy for you to validate it. And it'd be great if you can take a conditional knockout as well from us and validate that as well with the CRI. And finally, I don't want to leave you just saying that we are knockout or conditional knockout company. As I mentioned to you, we have a huge immuno-oncology portfolio and standing, especially in the humanized and immune checkpoint areas. So you can see here that we are actually world-class, and we're cut above the rest in terms of how we make our targets. We have single target, double target, triple target mice on dual backgrounds. So if someone wants to look at human PD-1 and human PD-1 ligand, we have that as well. If you take it further and look at those two genes and humanized version with a CTLA-4, we have that as well. So a very unique capability in all facets of mouse research and preclinical research, only here at GemPharmatech. That doesn't stop us there. We have a microbiome portfolio. We have a portfolio where we do preclinical studies, your pharmacokinetic studies, and we have disease models ranging from diabetes to cardiovascular to immuno-oncology. We can even do osteoarthritis, osteoporosis, bone disease. So there's no limit. We hope you can come to us, and we hope you can be that kid in that sweet shop mentality saying, you know what, I want this mice, I want to do this, I'm not sure. Well, you can, because you can say, now you can say, I can go to GemPharmatech. And we really want you to do your science, dream big, get whatever you want, come to us, and we'll provide it for you. So be that kid in that sweet shop, and hopefully we can communicate soon. So thank you very much for your participation. If you want to contact us, we are in the US and EU and also Asia as well. I'm going to leave this slide up so you can contact us. And please do email us at globalservice@gempharmatech.com. So thank you very much for your time, and we'll probably take some questions now.
Great, thank you so much, Pradeep. I love to be the kid in that sweet shop all over the world to save my trips. As people who joined us before, we have been running the KOAP in January this year, and this time around, Pradeep actually brought us more information and the updates that took place in the past several months. So as we wait for questions from this audience to come in, I do want to share one question that we have been receiving a lot from our customers regarding the KOAP portfolio, just to start the Q&A session. So this is a particular question regarding quality controls that we do on the mutant lines that we create through the KOAP project. Can you comment on the quality controls that we do on the KO or CKO mouse that we create? I think I can open this up to both Pradeep and Mark if you want to chime in.
So yeah, sure. We're very thorough in the quality control we do. As I showed you, we looked at SSH DNA. We look at repeats. We also do the validation in qPCR. So we don't give things out very easily, and we do thorough validation. And also, if the customer wants other things done, like sequencing, we can do that as well. So validation is a key to us, and we'll do that very thoroughly.
Great, thanks. I got one question. I think is a great question. Thank you for the person who asked. So it's a question from Mark, and it says, as a CEO of a company providing mice for research, where do you see the mouse business will go in the next few years? How would you like GemPharmatech to participate in this activity?
Well, thanks very much for that question. It's a great question. And as I say, I think that's why I'm more excited about the utility of mouse research now, even than when I started this more than 20 years ago. The dream was, and Oliver Smithies really was essential in starting this. And Pradeep showed a photo of Oliver, who no longer is with us, but an amazing, engaging scientist who sort of hit his third stride in his 70s and won a Nobel Prize. And Oliver's vision really was the modification, early stages of gene therapy, long before anyone thought about it, and being able to do the genetic modification in cell lines originally to study cancer, and then on to do those modifications in mice. And now that technology is there where we can really make any type of point mutation, humanization that we always dreamed of. And we don't have to just have random transgenesis. We can put genes into a safe harbor locus so we have consistent expression, and we can swap, mix and match the vast number of different human disease alleles that are coming out. So as I said, this is an exciting time to be a scientist, and the mice have been of never greater utility. And as you saw on the immuno-oncology, where we have double, triple mutants, we're only limited by our imagination of making better and better mouse models in almost any disease area that you're studying.
All right. I see that we're almost near the end of the webinar. I'll post this last question. Could you share some technical details about how you produce CKO by CRISPR? Do you have?
I can answer that because it's one of the few technical questions I can still answer these days. So yes, thanks for that question. Is that the vast majority of the CKOs, similar to what we did with the Knockout Mouse project, we choose a critical exon, or if we can't identify a critical exon because the gene is so novel, we knock out multiple exons. And in these cases too, we insert at two different sites, the LOXP sites. So we're not making a very long site, which might be difficult to get homologous recombination, but making two small insertions where they would be necessary. And that's important because in some cases, it might be a single exon that's relatively close together, but we have some that might be 10 kilobases away. So that would be much more difficult with homologous recombination. So we make two inserts of the LOXP site.
Mm-hmm. So the question actually continues. It says, do you use single-stranded or double-stranded guide?
In most cases, we're using single-stranded guides.
Mm-hmm. And how do you select your guide RNAs?
As everyone says, we have a computer.
There we go. Yeah, well, we are on the hour for this webinar. Thank you so much for all the participants and also our speakers, and hope to see you next time, and enjoy your day. Thank you.
Thanks, everyone. Thanks for joining.